Dissolution method development for fixed dose combinations, that is FDC drug products, is very challenging and critical. I welcome all the viewers to this informative video on the present topic of dissolution method development. See, the fixed dose formulations are those formulations which contain more than one active substance or more than one drug. It may contain 2 or 3 or sometimes more APIs or drug substances. And to develop a dissolution method for this type of formulation is challenging. Fixed dose formulations or fixed dose combination drug products are the products which combine 2 or more APIs in the single dosage form. Dissolution testing is required as a in vitro testing to know the in vivo behavior. Dissolution testing matters because it is required to ensure drug release and the product performance. It is critical for the quality control and the regulatory compliance. Dissolution testing is a performance testing. Also, it is required to meet the quality requirements. The goal in the FDC method development is to achieve accurate, efficient, and discriminatory dissolution testing. As we know that dissolution testing or method should be robust, should have sufficient discriminatory power, and should be simple, and the specifications are required to be finalized based on the dissolution of BE batches or clinical batches. As more than one API is present in the formulation, it becomes difficult to develop a suitable dissolution method. Generally, single dissolution method is advised or required, but it has some challenges. Now we will see why the single method is desirable, as it can simplify the routine QC and stability testing, because if you are using separate dissolution method for each of the API, then it will consume cost and time. Single dissolution method can reduce analytical workload and the cost, and single dissolution method has the advantage of enhancement of the lab output and efficiency of the chemists. Developing a single dissolution method with robustness and the required discriminatory power is very challenging, as the FDC contains the multiple APIs. These APIs have different physicochemical properties, different dose, and different solubilities. First is selecting the detection method. So you can use UV visible or HPLC based method. UV visible challenges like wavelength overlap among the components, poor specificity for complex mixture. These are the challenges. Then HPLC have some advantages over UV. Isocratic HPLC can provide you simple and reproducible results. Gradient HPLC is suitable for separating multiple APIs, reducing the runtime. Then another factor we need to consider in FDCs is large dose disparity. One API will be higher dose, another may be lower dose or ultra lower dose. For example, 1000 mg versus 5 mg or 1000 mg versus 10 mg. So these dose differences require dual detection method. Sometimes specific detectors are required, and the alignment is required for the detection. Then challenge is the ensuring discriminatory power for both the active substances. Purpose of discriminatory method is to detect changes in the formulation or process, and the changes are made in the range of 10 to 20%, and sometimes these changes are made by making the mismanufactured batch or bad batch. If the fixed dose combination contains soluble APIs, then it can be easy to develop the method. But if the combination product contains insoluble APIs belonging to BCS class 2 or 4, then the focus will be on discrimination mainly, and focus on dissolution, as bioavailability is dissolution rate limited for the low soluble APIs belonging to BCS class 2 and 4. There may be interaction of the APIs in the FDCs. There may be interaction of the excipients with APIs. Drug-drug interaction may be there, drug excipient interaction may be there, also excipient-excipient interaction may be there. So these interactions can significantly influence the drug release from the fixed dose combination formulations. Also, if the fixed dose combination is available in different release mechanisms, so managing the mixed release mechanism becomes very important and critical. If the formulation contains immediate release dose and extended release dose in a combination in the one FDC product like bilayer tablet with one layer as a sustained release or extended release or modified release and another layer as a immediate release, then it will require separate dissolution conditions. Separate dissolution methods are required to be developed. These type of dual release mechanism products require separate evaluation strategies. Recommended approach for this type of formulations is to use distinct dissolution methods so that you can justify the discrimination. Also, it is recommended that accurately assess the immediate and extended release performance of the formulation. Then dissolution specifications for the FDCs are given based on the pivotal batches, clinical batches using the dissolution data of 12 units. Selecting time points for dual release type of FDCs is like for IR component, you can provide specification at 15 minutes, 30 minutes or 45 minutes, and for ER component you can do sampling in 1 hour, 2 hour, 4 hour, 6 hour, 8 hour, up to 12 hour or 24 hour depending on to the release, and minimum 3 time point specifications are required to be given. Then how we will start for the method development if the product is FDC? Then the starting point is like use the established methods. There might be monographs available for the individual formulation in the USP, in BP, it may be in the literature, it may be there in the FDA database like OGD. So you can have a reference of individual API methods and you can combine them. Regulatory alignment improves development speed and compliance. So if you are using those methods, you can try with the existing methods, then you can make the necessary changes as per the formulation, and then you can set the dissolution method for FDCs. FDC product dissolution method development must balance the challenges, complexity and the efficiency. If 3 components are there, for example, and you are using separate dissolution methods, then every time for every batch 3 dissolutions are required to be done. Also 3 dissolutions will be required on the stability samples. So it will become very difficult. Also there are chances for the failure. So whenever you are working on the FDC's method development, understand the sync conditions, understand the release mechanism of the formulation, understand the BCS class, and then you can go for the method development. Always do the literature search, literature survey to come out with the specific single API method available for the simple type of formulation, and then you can start working onto the FDC. Then selection of the detector detection method, robustness and discrimination are the key points in the FDC dissolution method development. Always thoughtful planning and method validation ensures success in regulatory submission and the routine QC. Because once the method is finalized in the R&D, it will go to the QC testing lab, which will check the quality of the FDC and will release the formulation batches into the market. This is the overview of dissolution method development for the fixed dose formulation, fixed dose combination products or combination formulations. You can use the knowledge of single API formulation for the combination formulation, but always understand regarding the product design, immediate release part, extended release part, or sometime the API may be in the coating and one API may be in the cover of the tablet. Sometimes the capsule may contain pellets with different release profiles, so that is also required to be considered. Based on the experience, based on the knowledge, and based on the pharmacopoeial methods, you can start development of the dissolution method for FDCs. Thank you for watching the video.